C4 mechanism or Hatch and Slack cycle. Before we discussing the Hatch and Slack cycle, we can go for one more thing, that is photorespiration. What is meant by photorespiration? It's an abnormal reaction. We can say it's an abnormal reaction occur in C3 plants. So as I told you earlier, the Rubisco enzyme can catalyze both oxygenation and carboxylation reaction. So if the RBP combined with the oxygen affect the rate of photosynthesis. Okay, so that will affect the rate of photosynthesis. Plants need carbon dioxide. Okay, so in high temperature and oxygen concentration, Rubisco catalyze oxygenation of RBP rather than carboxylation. Okay, so that means the enzyme is not efficient. Okay, okay. If oxygenation enter into another set of reaction called photorespiration instead of Calvin cycle. Okay, so that is what here. So oxygenation, the plant actually need carboxylation rather than oxygenation. So oxygenation is not good for the productivity. Okay, so here another set of reaction will occur. So that is what we termed it as photorespiration. Okay, so so during that process, the plant taken in oxygen and release of carbon dioxide occur. Carbon dioxide is released. Okay, and oxygen is received. So it occur in the presence of light. So that is we termed it as photorespiration. Photo means light. Respiration like phenomenon. Not actually. So this is not a respiration, but actually it appear like respiration because it during this process oxygen is taken in and carbon dioxide is released. Okay, so so that we termed it as photorespiration. Okay, so here we can otherwise we termed it as C2 cycle, photosynthetic carbon oxidation cycle. All are same. Glycolate cycle. Okay, this abnormal reaction was discovered by two scientists, Dicker and Teo. Okay, so we can just to go through that process. It starts from chloroplast. From chloroplast that process start. Okay, and it needs two other organelles, peroxisome and mitochondria. So all these three organelles need together, all of them together need required to complete the reaction. So in the chloroplast, the first step, the combination of RBP with oxygenation can be seen. So RBP combined with oxygen, that means 5 carbon compound combined with a oxygen, 5 carbon compound. The RBP is 5 carbon, we know that it associate with oxygen, so the number of carbon there is no change. Okay, so and then what so that is the first step. Okay, so here oxygenation. So after result, two compounds will be produced. After that, one 3 carbon compound and one 2 carbon compound will be produced. The 3 carbon compound is known as phosphoglyceric acid and 2 carbon compound is phosphoglycolate. So these are the two compounds produced after the oxygenation of RBP. Okay, so the phosphoglyceric acid can enter into Calvin cycle and can produce glucose. Okay, but the second compound is abnormal compound, so that cannot enter into Calvin cycle. Okay, so here so the glucose production will be affected. So then in the next step, the phosphoglycolate remove one phosphorus. Phosphoglycolate is the compound here, we know that. So the phosphoglycolate, from this phosphoglycolate one phosphorus is removed. Okay, so so that we can see here. So as a result, a compound known as glycolate will be produced in the chloroplast itself. Glycolate enter into the second organelle, so that is peroxisome. Okay, here the glycolate combine with oxygen and oxygenation. Here again one oxygenation to form glyoxylate and hydrogen peroxide. Glyoxylate and hydrogen peroxide are produced. Okay, so hydrogen, so the glyoxylate receive one amino group from where, from glutamic acid. It receive one amino group, so and it become glycine. Glyoxylate get converted into an amino acid known as glycine. Okay, glycine. So the glycine will be produced, and then glycine in the next step enter into the mitochondria. Next cell organelle is mitochondria. Okay, so here this glycine, we know that all these are 2-carbon compound. Glycine is a 2-carbon amino acid. Okay, so here this amino acid is produced continuously. Which amino acid? Glycine. As a result of this reaction, this glycine continuously produced and enter into the mitochondria. After some time, 2 glycine molecule together combine to form serine, another amino acid converted into serine. Okay, so glycine 2-carbon compound. So 2 carbon compound combines with and produce a 3-carbon compound known as serine. During that process, 1 carbon is released in the form of carbon dioxide. Okay, 1 carbon is released in the form of carbon dioxide. Okay, along with that an oxidation also will occur. So thus NAD become NADH2. NAD receive the hydrogen ion and electron to become NADH2. So amino group, one amino group is also released. So this is a complex set of reaction, oxidative decarboxylation we can say. Okay, oxidation, removal. So here NAD get converted to NADH2. Carbon dioxide is removed, and an amino group also released from that reaction. So the 3-carbon amino acid serine will be produced. Okay. So serine then in the next step enter into the peroxisome. Again come back to the organelle peroxisome. Serine now enter into the peroxisome. Serine again receive again donate the amino acid to alpha ketoglutaric acid. Alpha ketoglutaric acid receive the amino group from serine. Serine is an amino acid, we know that. So that release the amino group, donate the amino group to alpha ketoglutaric acid. Okay, so as a result, serine get converted into hydroxypyruvate. Okay. Okay, hydroxypyruvate in the next step get converted to glycerate by reduction. Hydroxypyruvate is reduced to glycerate. Here the for that reduction purpose, one NADH2 molecule is utilized. NADH2 donate the hydrogen ion and electron for that purpose. So glycerate in the last step again come back to the chloroplast glycerate and get converted to phosphoglyceric acid by receiving 1 ATP molecule. So that is phosphoglyceric acid can again enter into the Calvin cycle. So here this is a series of reaction occur, we can see, as a result of oxygenation of REBB in 3-cell organelle. So during that process we can see there is no net energy production, but 1 ATP lost during this process. Energy loss is there. Okay. So the here one NADH2 produced, here one NADH2 consumed here in this reaction. Okay, 0. So net 0 change. Okay, so one ATP lost here, then one carbon dioxide is removed. Instead of fixing carbon dioxide, one carbon dioxide removed. Okay, in overall, most of the scientists considered this is an abnormal set of reaction, but there are some other arguments are also there. That means some amino acids are produced that are useful for the cell. Okay, anyway, okay, so that is what about photorespiration. So this is not a favorable thing, we can say. So it occur this C3 cycle. Most of the plants may face this situation in high temperature and oxygen concentration. When the oxygen concentration increases, there is more chance of oxygenation. Okay. So more oxygen molecule will combine with the REBP. Okay, so plants need to overcome this. It will reduce the efficiency of photosynthesis. Okay. Anyway, some plants developed adaptations to overcome photorespiration. Okay. So for that purpose, there is another mechanism evolved. So that is C4 mechanism. This mechanism is usually present in tropical plants, especially grasses. So means those plants living at high temperature, moderately high temperature in tropical climate. Okay, so so there is more chance of photorespiration. So to overcome that, this new mechanism developed. Okay, so it can be seen mainly in plants like sugarcane, maize, sorghum, amaranthus, millets, etc. Right. So this process can also be termed Hatch-Chance like cycle, dicarboxylic acid cycle, etc. So we can go through that process. Okay, so this is the mechanism here. The plants showing this type of special type of photosynthesis developed a special type of modifications in their leaf. Okay, so they we can see, if we take a section of the leaf, a special type of anatomy can be seen here. So here at the center I just represented the vascular bundle, xylem and phloem, and surrounding that a few layers of bundle sheath cells are there. Okay, bundle sheath cells represented here in that blue color. The bundle sheath cells are very compactly arranged. You can see here these cells are very tightly packed close to each other, so that there is no intercellular spaces in between, and one or two or three layers of these bundle sheath cells can be seen like this, arranged in a ring fashion. Okay, and their cell walls are thick and impervious to gases, so it prevents the entry of gases, does not permit the passage of gases into that. Okay, so this region is totally airtight. Okay, so the atmospheric gases cannot pass into this or enter into this bundle sheath cells. Okay, so that is one of the important thing. And another thing is that, so here the chloroplasts are large in size. Okay, large chloroplast you can see almost arranged in a concentric manner, and they are without grana. These chloroplasts never develop grana. they are free. Thylakoid membranes are there, but there is no grana present in this agranal chloroplast. So that is the term that we usually used. Okay, large number of agranal chloroplast can be seen inside the bundle sheath cells. Okay, so then when we move on to the other region, the normal mesophyll cells you can see, and where the cells are loosely packed, arranged with a lot of intercellular spaces, and having normal chloroplast with grana we can see. And so that is what here we can see. So that is the general anatomy of this type of plants, category of plants. So here this anatomy, special type of anatomy, usually we termed it as Kranz anatomy. Okay, Kranz. Okay, Kranz is a word came from the German word. Kranz means ring or wreath, ring like or wreath like arrangement. Here can be seen here. So that is the special type of anatomy is important here in these plants. So so that is what we can see. So what is the mechanism actually? So here in REBP or and Rubisco enzyme, so important for Calvin cycle. So these enzymes are present only in the chloroplast of the bundle sheath cells. In the bundle sheath chloroplast only the Rubisco and REBP are present. Okay, in mesophyll cell this Rubisco enzyme is totally absent, and REBP, pentose sugar, REBP are also absent in the mesophyll cells. Okay, so the oxygen from the atmosphere can directly enter, oxygen, carbon dioxide, everything can enter into the mesophyll cells here and there, and it can diffuse anywhere else. Okay, oxygen can enter into these mesophyll cells. Okay, now there is no restriction. Okay, so the mesophyll cell contain mesophyll cell chloroplast contain one carbon dioxide acceptor, that is PEP. Okay, PEP is phosphoenol pyruvate. It's a 3-carbon compound. So instead of REBP, here PEP absorb the carbon dioxide in the mesophyll cells. Okay, and here the enzyme is PPase or Pepco enzyme, Pepco, phosphoenol pyruvate carboxylase enzyme. So that enzyme catalyzes the carboxylation. Okay, so what is the importance here? The PEP have no affinity for carbon oxygen, and this enzyme, the Pepco enzyme, never carry out oxygenation reaction. There is no attachment with oxygen. Okay, so there is no chance, oxygen never combines with the REBP, no chance for oxygenation. Okay, so 100% carboxylation, only carbon dioxide can combine with the PEP, so it can overcome avoid oxygenation process. Okay, because this enzyme is very efficient, and that lead to the formation of, so here if a PEP combined with carbon dioxide, PEP we know that is a 3-carbon compound. Here the carbon dioxide also combined with that. Okay, one more carbon. So here the 4-carbon compound, first stable compound is produced, that is oxaloacetic acid. So that is the 4-carbon compound. Okay, so 4 carbon, so that is what we termed it as C4 cycle. Okay, oxaloacetic acid immediately converted to malic acid or aspartic acid. Usually it converted to malic acid. So that it reduced to malic acid, actually it reduced to malic acid with the help of NADPH2. Okay, so reduced coenzyme donate the hydrogen ion and electron, thus it become malic acid. Malic acid, again the same 4-carbon compound will be produced in the mesophyll cell. Okay, so then the malic acid, 4-carbon malic acid, again so enter into the bundle sheaths through plasmodesmatal connections can be seen between the neighboring cells. and through that cells the movement of the malic acid occur. Malic acid gradually enter into the bundle sheath cells. Okay, inside the bundle sheath the malic acid undergo decarboxylation. That means carbon dioxide is removed, okay, given out. Carbon dioxide come out from malic acid, and a 3 carbon compound will be produced, pyruvate. Malic acid, remember, is a 4 carbon compound. Remove 1 carbon dioxide carbon and it become the 3 carbon pyruvate, right? So the pyruvate is produced. Okay, the carbon dioxide thus we can see released into the bundle sheath chloroplast, inside the chloroplast of the bundle sheath. So here we know that RUBP is there in that chloroplast, bundle sheath chloroplast. The carbon dioxide acceptor RUBP is already there, and also the Rubisco enzyme, Rubisco. Okay, so the carbon dioxide combined with the REBP, that is carboxylation, and here by with the help of the Rubisco enzyme, and that lead to the Calvin cycle. As a result, the dark reaction or C3 cycle will occur, and that lead to the formation of glucose. So here carbon dioxide only can combine with RB. Here there is no presence of oxygen. Okay, so that process, oxygenation, never occur. Why? Because there is no oxygen here. Okay, so the plant can overcome photorespiration. So there will be no photorespiration. No photorespiration will be there. Okay, yes. So that glucose will be produced here, and what will happen then? For pyruvic acid, the pyruvic acid 3 carbon compound produced here again enter into the mesophyll cells, returned back to the mesophyll cell. 3 carbon compound again converted to phosphoenol pyruvic acid. Okay, with the help of 2 ATP molecules will be there. So that lead to the formation of phosphoenol pyruvate. So that is the reaction. So here 2 ATP is required for this set of reaction. So here PEP absorb carbon dioxide and enter into the convert into malic acid, then enter into the mesophyll, then come back again. It converted into PEP. So this reaction, what is the importance or purpose of this reaction? Here the carbon dioxide is taken from the mesophyll and dumped into the bundle sheath. So the carbon dioxide is taken from outside and put it into the inside, taken, put, released here. So the carbon dioxide concentrating mechanism, carbon dioxide is pumped into the bundle sheath. So here in the bundle sheath the Calvin cycle will continue uninterrupted, producing glucose. So the plants can produce the glucose efficiently. Efficiency of photosynthesis increases because there is no photorespiration. Okay, so but what is the disadvantage for this cycle to operate? So this is the actual part of the Hatch-Like cycle, right? So to operate this much reaction, an expense 2 ATP need to be spent. The plant need to spend 2 ATP for one cycle. Okay, so we know that through Calvin cycle plants need 18 ATP and 12 NADPH to produce one glucose molecule. Okay, 12 NADP. So for to produce one glucose molecule 6 carbon is needed. 6 carbon is needed. For transporting 1 carbon from mesophyll to bundle sheath, 2 ATP is needed. So for to transport 6 time, for to operate this cycle 6 time, 12 NADP, 6 into 2, 12 ATP is needed. So there is an additional expense of 12 ATP for to complete the process, for to produce 1 glucose molecule. Okay, so 18 plus 12, 12 is additional requirement, extra needed 2 ATP for each carbon to enter the bundle sheath. So that 6 carbon means 12 ATP. Okay, so 30 ATP is needed, and there is no change of NADPH2. NADPH2 remains the same. Okay, so 30 ATP is needed for producing 1 glucose molecule in Hatch-like cycle or C4 mechanism. Okay, so even though the this mechanism is expensive, it is efficient. So that is the advantage for the plant. Okay, so that is the advantage. Even in extreme environmental condition, extreme weather, adverse environment, the plant can do photosynthesis efficiently. Okay, so that is the adaptation here. Okay, yes. C4 plants, so there are two type of carboxylation is there. First one carboxylation occur in the mesophyll cell, and the second one in the bundle sheath. Okay, so this is first primary carboxylation and secondary carboxylation. Okay, first carbon dioxide acceptor is PEP, and the enzyme is PEPCO or PEPase. Okay, the second enzyme is Rubisco, normal enzyme Rubisco, and a carbon acceptor is REBP. So there are two carboxylation reaction. Okay, so this is what given in our NCRT. This is the diagram given in our NCRT. Okay, so here carbon dioxide enter in the form of carbonic acid, PEP combined with that, fixation occur, C4 acid. C4 acid is actually oxaloacetic acid and malic acid transported here into the bundle sheath. Here decarboxylation occur for malic acid, so fixation through Kelvin cycle, then C3 acid, that is pyruvic acid, transported back into the mesophyll. Again phosphoenol pyruvate is regenerated. For that process here 2 ATP is needed. 2 ATP get converted to 2 ADP. Okay, so that is the overall process. You need to go through this diagram also. Okay, yes.